Okay, so we are now being recorded. So again, welcome to this ninth video teleconference for BIOL 105L YC. So in terms of our schedule, this is our calendar for April. We are currently in sync with the number of video teleconferences that we are supposed to be doing. However, if you notice, the schedule or the title of the activity is interchange. So this week we are working on exercise number 10 actually, no, and ELISA. And then next week we will be discussing about a topic that is not really included in our laboratory manual, and that's about chapter 16 on cancer. Okay, so, however, for next week, there is another conflict on my schedule for Thursday, Friday, and Saturday. That's why for your class, I am requesting that we do a video teleconference on April 20, still the same time, 2 to 3 p.m. And we can have a live discussion about chapter 16 or cancer during that video teleconference. So after next week's discussion on the extra topic on cancer, we're actually going to be done already with all of the laboratory activities. In fact, this is the last lab activity, and it's a virtual lab activity. So that on the last week of April, you're already free to do the final requirement for this course. No? So I will still have to give the final requirement by next week. So are there any concerns with regards to our schedule as shown here?
None, sir.
No, sir.
Okay, sige. So if there are none, this video teleconference is devoted to review, no, or also as a discussion on the lab activity that we are doing right now. Let me admit someone in first. Okay, and that is a virtual laboratory activity on ELISA, no, or ELISA. So this is enzyme-linked immunosorbent assay. It's a virtual laboratory activity. So I hope all of you here have already finished, no, and are just making your lab reports for this. So the lab report is still the usual lab report. No, I received many questions about that. But for answering the questions, no need to answer the question in the lab manual about HIV because that's not really the one performed. So download the worksheet here instead, and this will be the one that you will be answering or including in your lab report. So we will proceed with a discussion on ELISA. Okay, so in the virtual laboratory, ELISA was used to test, if the patient has a particular disease, and that disease is an autoimmune disease in the lab, in the virtual lab activity. So the systemic lupus erythematosus, if I'm not mistaken. So it's an autoimmune disease wherein the body's own immune system is attacking the body's own cell. So we will not cover much about that because you've done that in your lab activity. No? So instead, I'll be discussing here the principle. So just to reiterate it again. So our bodies, animals' bodies produce antibodies. And antibodies are produced by our immune system when a specific antigen or a foreign body is detected in the immune system. So what are these foreign bodies? They can be bacteria, They can be allergens like pollen grains, or they can be viruses, like the SARS-CoV-2 that we are aware of right now. So when these antigens enter our body, our immune system, a portion of it called the adaptive immune system, makes antibodies to attack the antigen. Okay, and these antibodies are very specific. So, for example, if the antigen that entered your body is a SARS-CoV-2 virus, then your body will make an antibody only for the SARS-CoV-2 virus. If another virus, say SARS-CoV-3, okay, will invade your body, then your body will not be able to do anything about that yet because you still haven't created the antibodies for that new virus. Okay, so this is very specific and it binds to the antigen. Um, in ELISA, or enzyme-linked immunosorbent assay, the goal is to determine and measure whether you have a specific antibody. So if it's found out that you have an antibody for SARS-CoV-2, that means you have been sick or you are still sick with COVID-19 because you have the antibody for SARS-CoV-2. Okay, so one way to find out if you have those antibodies is through ELISA, the activity that you did in the virtual lab. And usually, the antibodies are obtained from the patient's blood, particularly in the serum portion of the blood. So in this particular video teleconference, I'd like us to focus on another situation wherein ELISA is actually applied, and that's in the rapid test kits for COVID-19. So if you're not familiar yet with this or you haven't used one, it would most likely look like this. No, there is this strip, para siyang pregnancy test strip. Then there is this portion where the blood from the patient is dropped or inserted or inputted. And then there are these strips, M, G, and C. C stands for control. So it should always turn out red whether you have the disease or not. And then G and M stand for the type of antibody. So kaning IgM and IgG are antibodies. Let me admit someone in again. So IgG and IgM are actually antibodies, right? So in this second test here, we see that the control is red, so there's no problem with that. But notice this, the antibody M, IgM, is red. So meaning to say, we can say that this patient has an antibody against COVID-19. Therefore, he or she must be positive for the disease. The same is true for patient number 3 here, where the IgG is red. And of course, if IgG and IgM are both red in the test, then we are very sure that you are positive with the disease. So this is rapid test because it can be done in a matter of minutes. You will see the result. So why am I showing this to you? Because the principle of this is ELISA, similar to ELISA, the presence of test for the presence of antibodies. So that's rapid test kit. Okay. Now, before we discuss the steps in ELISA, I would want us to go into sort of a mini lesson on SARS-CoV-2, the disease-causing agent of COVID-19. Who among you here are familiar with the virus SARS-CoV-2? Have you read about it? Are you familiar on how it works? Anyone? Silence, I think, means not really. Okay, so perhaps because this wasn't really part of any of our lessons before, maybe you did not have time to read about SARS-CoV-2 or how it works, no? Or maybe some of you do, but you're just not so sure. So don't worry, in this lecture or in this discussion, I will show that to you. Okay, so this is actually a lecture that I prepared for Tiger Talks. If you're familiar with the Festival of Artsies, there was these Tiger Talks. And one of the talks was about SARS-CoV-2 and vaccines. So these are my slides. So this one is a model of SARS-CoV-2, and it's a coronavirus because it looks like a sphere with corona surrounding it. So just like the sun's corona, no? So na mga rays. And those corona are actually known as spike proteins. So if we try to see the parts of the virus, there is this membrane, okay? And this is actually made up of the envelope protein or the E protein. And then interspersed throughout the membrane will be sort of integral proteins known as membrane proteins. And then also still on this envelope protein are these triangular shaped structures that I have here. And these are known as spike proteins. And inside this capsid, no, this is the capsid of the virus, the protein. Inside are special proteins which actually help stabilize the RNA of the virus. These are known as nucleocapsid proteins. And then you have your RNA. So SARS-CoV-2 is an RNA virus. Okay, it's an RNA virus. So this is now our structure for SARS-CoV-2. And the spike protein is of very significant use, no, or significant importance in dealing with this type of virus because this is the protein that is used by the virus to enter our cells. So here's a picture of SARS-CoV-2, and indeed they look like little suns, little deadly suns. So we have here the spherical virus, and then we have the spike proteins as seen by these bluish corona outside of the virus. And here we can just see a cell, and then here's a virus that has started attaching to the cell. No? Okay, so in the next few slides, we will see which cells are the targets. Okay, but before I go to that, does any one of you know the target cells of the SARS-CoV-2 virus? Give me examples of cells na target or are affected.
Lungs, sir?
Okay, the cells in the lungs. Okay, so that's correct, diba? In fact, the first symptoms are related to our respiratory system, diba? Kanang sipunon, ubuhon, maglisod, o ginhawa. Okay, so you're correct. So this one, the illustration here is the surface of a cell, no? And these sticks that I've drawn here, these are actually integral proteins, okay? And then also on the surface of this cell are these receptors, okay, these green ones. What are these? Okay, these are angiotensin converting enzyme 2. Okay, so these are enzymes. So the name is ACE, no? Human ACE 2. Angiotensin converting enzyme 2. Have you heard about this from the news? If you haven't, then that's not a problem at all. So the question is, what's the function of this? Okay. The human angiotensin converting enzyme 2, as the name implies, converts angiotensin 1 into angiotensin 2. Sama ng angiotensin. Angio meaning blood vessel, something related to blood vessel. Tensin, something related to tensing, tense. Okay, now, angiotensin 1 is converted to angiotensin 2 by this enzyme. Angiotensin 1 is a vasoconstrictor. What is a vasoconstrictor? Anyone wants to guess? Unsa ng vasoconstrictor? Vasovascular, something related to blood vessels. So, unsa man ang vasoconstrictor?
Tig-contract, sir. Pag naro sa blood vessel.
Exactly, no? So that's angiotensin 1. Now, if you're a person who is hypertensive, you might have a lot of angiotensin 1. And you don't want angiotensin 1 present in excess in your body. Okay, because they constrict the blood vessel and therefore your blood pressure will increase. Okay, so the work of the angiotensin converting enzyme 2 is to convert the angiotensin 1 into angiotensin 2. And angiotensin 2 has the opposite effect from angiotensin 1. It is a vasodilator. Okay, so ang vasodilator, instead of constricting your blood vessel, it will make the pathway in the blood vessel bigger by relaxing the muscle around the blood vessel. So again, the work of the ACE2 convert angiotensin 1, which is a vasoconstrictor, into angiotensin 2, which is a vasodilator. So that's a good work, no? And these are found in the cells in your lungs and lining your respiratory system, the cells lining your blood vessels, di ba? The cells in your kidneys, and the cells in your intestines. Okay? So these are very important, kaning ACE2, no, on the cells because they convert angiotensin 1 to angiotensin 2. Unfortunately, the SARS-CoV-2's spike protein interrupts with the ACE2. Now look at this, it binds, and then here's another one. It binds with the ACE2. As a result, ang ACE2 dili na makatrabaho. No? So the ACE2 is preoccupied with the spike proteins of the coronavirus. And so, what do you think will happen to people who are hypertensive if there is SARS-CoV-2 in their body? Unsa may mahitabo?
Mas masamot, sir.
All right, 'di ba? Mao nang if you've heard or if you listen to the news, 'di ba? They usually mention elderly people or young very young people, babies as having more tendency of dying when they contract the SARS-CoV-2 virus, no? And one of the things that they mention is those people who are hypertensive, no? Hypertension is a comorbidity to COVID-19. Why? Because the SARS-CoV-2 virus interacts with these ACE2, which are supposed to be working to convert angiotensin 1 to angiotensin 2. So karon dili na siya maka-work and musamot ang um, hypertension sa tawo nga hypertensive, no, when there is this SARS-CoV-2 interrupting. So now that the spike proteins are bound, the SARS-CoV-2 may then combine with the cell's membrane and it will open a pathway that will allow the RNA of the virus to go into the cell. So, makasulod din ang RNA into the cell. And of course, we know this is an mRNA. So the cell's ribosomes, now these are the genes of the virus, are going to read this mRNA to produce proteins. So here you have the nucleocapsid proteins, then you have the envelope proteins, then you have the membrane proteins, and then you have the spike proteins made by the host cell's own ribosomes. And once these are made, they can then self-assemble into a new virus. Okay? So this new virus will then be free to go out of the cell. The cell will die, no? The cell will lyse. The virus will go out of the cell and it can now infect other cells. So that's a background on how SARS-CoV-2 infects specific cells in our bodies. No, those that are lining our respiratory system are most affected by this. Okay, so what about our immune system? So our immune system is made of two parts, and don't worry, this is a topic in our lecture, no, later on. This is just an overview. Don't worry, I will not be discussing a lot of things. Our immune system has this adaptive immunity portion, not only for vertebrates. Okay. First, there is this humoral response, meaning to say, if the virus enters your body, your humoral immune system will produce antibodies, no, the B cells, to trap them. So look at this. The antibodies will surround the virus, the antigen. Then there is also this cell-mediated response wherein if the virus has already entered your cells, then it might be too late for the antibodies to go inside the cell, of course. So what the host cell does is it removes a portion of the spike protein and it presents it to the outside so that it can give a signal that it is infected. Okay, so I'm infected. Kill me. That's what the infected host cell will tell the natural killer cell, the cytotoxic T cells. Okay, so the cytotoxic T cells will detect this and they will release an enzyme that will cause the infected host cell to die, to burst. And once this is burst open, the viruses that are inside will be freed outside and the antibodies will be able to um, uh, trap them, to bind to them. Okay, so this is the importance of the antibody now in case of SARS-CoV-2. So balik tayo sa ELISA. ELISA's principle is to determine and measure whether an antibody is present in your blood. Okay? In the case of SARS-CoV-2, if there's an antibody, then you have the virus. In the case of AIDS, if you have the antibody for HIV, then most likely you have AIDS or HIV also in your body. No? So, mo na ang ELISA. So, what are the steps? Before I talk about the steps, are there any questions or concerns? You're free to ask, no? Especially that I'm talking about a new topic here that might interest you. Were you able to connect ELISA with what I was talking about here? Kay basig sa man ni sister nagstorya na mag COVID-19 nga ELISA man tamong topic. Any questions, concerns, comments? There is something that you didn't understand or you're curious about, you ask. Okay, it's okay. Wala. Okay, so let's proceed then to the steps of performing ELISA, which I'm sure you're familiar already because you did your virtual lab activity. Okay. So if you're familiar, let me ask you, what's the first step? Unsa may first step sa ELISA? What will you do first?
Centrifuging the sample, sir.
Okay, centrifuge. Okay, all right, of course, no. Thank you for that. That's correct. But let's skip that one. Once you have your sample ready now, what should be placed first? Is it the antigen or the antibody? But you have the microtiter plates. What should be placed first? The antigen or the antibody?
The antigen, sir.
The antigen. Yes, that's correct. Okay. So the antigen needs to be placed in first. So look at this illustration. Okay. So this is a surface of your microtiter plate. So here is an antigen. No? So kung SARS-CoV-2 or COVID-19 pang gitest dire, this antigen here will be a portion of the spike protein of the SARS-CoV-2. If you are testing for AIDS, no, or HIV, then this antigen that will be placed here will be a portion of the virus that causes AIDS, no, so human immunodeficiency virus. If you are testing for SLS, systemic lupus erythematosus, then this one here, this antigen, will be a portion of the cell that contains the signals, or it could be your own cell because your own cell has antigen. No? Your own cells already have their own antigens. And remember, in SLE, it is an autoimmune disease. So we are testing your immune system against your own cells. So the antigen here should be your own cell, no, if it's SLE. So this is the microtiter plate and samples are placed here. So if you cannot see it, there are actually letters here, A, B, C, D, E, until H. And then there are also numbers. So they are placed there so that the scientist working or the lab technician that's working will not be lost, no? Para dili mag-mix up ang samples. And of course, you use pipetting techniques to dispense the samples here. Okay. And once the samples are dispensed, okay, those samples from the blood will actually contain an antibody. Now, if the patient is positive for the disease or for SLE, then that sample of a blood should contain an antibody. The antibody will stick to the antigen as shown here. So kaning mga Y-shaped objects are the antibodies. So dire pa lang daan nga illustration, we can already see that this is positive. But in the laboratory, you will see no obvious reactions. No NVR, no visible reactions. So what really is needed is a secondary antibody. Okay. So unsa maning secondary antibody, where is it from? Is it still from you or where is it from? Is this from the patient or is this from somewhere else?
Lahi na, sir.
Okay. So this is not anymore from the patient, no? So take note na the antibody will stick to the antigen. No, because it recognizes this as a foreign body, so it will stick to that. So kaning secondary antibody should not be from the same patient. These will not react with the primary antibodies. So the secondary antibody will stick to the primary antibodies because they are also foreign to the secondary antibody. So you said no. So what are the probable sources of the secondary antibodies? Dili makagikan sa patient, ahaman siya gikan.
Rabbit, sir.
All right. So it's from another mammal, no, like a rabbit. So, can you imagine? Imagine niyo, no? So, ang antibody sa rabbit, ibutang sa imong antibody, of course, it will stick because they are foreign to each other. No, so you get uh, the idea there. The antibody should be foreign to the secondary antibody, so from a different animal source. But not only that, notice this: the secondary antibodies also contain an enzyme. Kinsa may nagbutangan ng enzyme, sir, gibutang na daan sa scientist sa secondary antibody, iyang gi-connect. Okay? So meaning to say, this is pre-made in the laboratory, this secondary antibody. So there's an enzyme, usually a peroxidase, no, from the horseradish, and it's stuck to the secondary antibody. So after the secondary antibody is added, it will be washed. Okay. Before I continue, I missed something here. In adding the primary antibody from the patient, after that, it should be washed also, no, so that the antibodies that did not stick to the antigen will be washed away. So only those that stuck will stay, no? And then same is true here. Wash, and then only those that are stuck will remain. What's the next step? Now that you have your secondary antibody with the enzyme.
Incubate, sir.
Incubate, okay? After incubation, what's the purpose of your, you mentioned incubation, what's the purpose for incubation?
Para katong ang enzyme, sir, kay mag-attach sila sa substrate.
Okay, and the substrate being the primary antibody. I think that's what you meant. Okay, so sige, let's go beyond that one now. Now that we have the secondary antibody with the enzymes attached, no, okay, so let's say delay incubation ang answer, no? What do we do next in ELISA? Kung saan itong signal nga, oy, positive ka for this disease. What needs to be added next?
Substrate, sir.
All right, the substrate for the enzyme. Kay enzyme man, di ba? This is an enzyme-linked antibody. Mao na ang tawag ELISA, enzyme-linked immune sorbent assay. The substrate, meaning to say, the chemical that will react because the enzymes are present. And that chemical is usually in the form of a chromogen. Okay? A chromogen is a chemical that will change in color once it reacts with the help of the enzyme, no, because there's an enzyme. So here, the chemical or the substrate is added. It's colorless at first, no? Look at these colorless circles. But once it interacts with the enzymes from the secondary antibody, it will react and change color into yellow. No? So that's why this example is from the SLE example wherein it turned yellow. In the HIV test, the one in your lab manual, it will turn pinkish. Okay, the chromogen will turn pinkish. So chromogen means color former, no? Color former. So chromogen. So now, pag nag-change ang color, meaning to say you are positive for the antibody, and therefore you must have the disease. Okay? So nagets ninyo how ELISA will detect if you have the disease or not. Okay? So again, no, review. Na lang. Here's a review of ELISA. So first, there is an antigen, no, the causative agent of the disease. So for example, this can be SARS-CoV-2 spike protein. Next, ikaw, gikuhaan kag dugo. Your blood will be placed on the antigen. If you have antibodies in your blood that are specific to the antigen na SARS-CoV-2, they will stick on the SARS-CoV-2 antigen. Next, this will be washed. Only those that will stick will stay. Next, a secondary antibody for your primary antibody will be added. And if your primary antibody is stuck there, the secondary antibody will also get stuck. What's more is they contain an enzyme, no, a peroxidase enzyme. And then lastly, a chromogen will be added. And the chromogen will react with the enzyme from the secondary antibody and will change color. And meaning to say that you have the antibody for SARS-CoV-2 and that you have COVID-19. Okay? So as shown here in these strips. So kani natawo kaning first negative kay ang control raman ang nag-red. Okay, so negative. But these people, second, third, and fourth, are positive for the presence of the immunoglobulin M and G, which are antibodies, no, for COVID-19. So they are positive. Okay, so any questions about ELISA and about this fast test kit? Questions, concerns? Nakasabot ra mo? Who understood? Say me, sir, or me. Wala yung nakasabot? If you understood, say I did.
Sir, nakoy question.
Okay, sige.
Like, nag-read na ko ang why nga part sa simulation, sir, pero wala gyapon ko kasabot. Ang ngano kailangan pa nga i-wash more than 3 times ang katong sample with PBS?
The sample that was, what do you mean? Which sample are you talking about? Sample blood from the patient?
Taning sa remove the fluid from each well with the pipettor and wash with 0.1 ml of PBS.
Okay. Remove the fluid from each pipettor. Sama na siya nga portion. The washing, what I can tell you here though is the purpose of the washing, if this is the washing that you're talking about, is to make sure that only antibodies that got stuck to your antigen remains, no? Otherwise, if nagbutang kag sample, then wala nimo siya gi-wash. You have other antibodies also for other diseases, for other pathogens. So, dili na din specific ang resulta sa ELISA kung nagsagol-sagol ang antibodies. Now, so mao nang kinahanglan i-wash para the other antibodies that are not stuck here will be washed away. So that's what I can say about that.
Okay, sir. Thank you.
So what about the others?
Nakoy another question, sir.
Sige.
Di ba, na-read man gud ako before na ang rapid tests kay dili sila always ga-give og correct na result. So does that mean nga since similar man sila sa ELISA, does that mean nga ang ELISA kay dili pod ga give og correct nga results?
Hmm. Um, Not necessarily that doesn't give a correct result, but could be. Thank you for the question. That's a good question. It could be false negative or false positive. No, that's what you're talking about. And usually, Manggod, let me clarify. ELISA and this rapid test are the same in principle. No, but for ELISA, um, bale under ELISA will be this rapid test. ELISA has many uses. For example, katong SLE, no, diagnosis for autoimmune diseases. Ang kani magong COVID-19 nga mag-diagnose ka if you have COVID-19 or not. It is much more urgent, di ba? Kung gusto ka mahibalaan mo kung nakay COVID-19 or wala. So that's where the problem in the use of rapid tests come in. Now, not necessarily that ELISA or rapid testing is not a reliable method, but it's somewhere on the urgency of diagnosing if you have COVID-19 or wala. Okay, so good question. And this will lead me to ask you, okay, what do you think are the instances that can contribute to a false negative? Sa manang false negative, abi ni mo wala siya sakit kay iyang resulta kaning una, pero naadi siya COVID-19. Can anyone guess or give an answer? Now, negative yung result, pero naadi siya sakit. Gamay ra ang iyang antibodies. Correct. Correct. No? So it's, not all of us have equal immune systems. Okay? So there are some people, especially those that are immunodeficient, kanang ilang immune system galisod og produce og antibodies, no? Nga naapekto na sila SARS-CoV-2 daghan kaayo, klaro na kay gaubo-ubo or gasipon-sipon, pero ang ilang body wala pa nakaproduce og antibodies. So pag test sa ila aning rapid test kit, walay nang gawas nga positive sila ani nga antibody. So that is correct. Very good. There is still another instance. Can anyone tell us, aside from having a weak immune system, unsa pay other cause of false negative? Let's say naglaag-laag ko this morning, no, and then I forgot to wear my mask niadto ko sa kugon, then dito di ay naka-contract na ko og SARS-CoV-2, then just now nagpa-rapid test ko, ang nigawa sa akong test negative. Why?
The person is still in the window period, sir.
Okay, so there's an element of time, no, if that's what you meant. For example, in my example, no, I just contracted the virus this morning. No, dayon nagpa-test dayon ko in my blood if I have the antibodies against the virus. Of course, my immune system is still starting to produce antibodies. So pwedeng gamay pa kayong antibodies, wala pa na detect diri sa rapid test. So false negative because after a week, no, or later on, sip uno na din ko maglisod na ko ginhawa kay naa na day ko yung COVID-19, then in my test nga bag-o pa ko nakakuha sa COVID-19, wala pa ko yung antibodies na produce against COVID-19. So that's false negative. Now let's go to the opposite situation, false positive. Meaning to say, I get all the results here, the three results here on the right, pero wala day ko'y sakit. What are the reasons for that? Naulian na. Okay, correct. Okay. So, let me elaborate on that no, pero correct to siya. That's very correct. If I got the virus, kung nasakit ko last month, naayon ako, the antibodies that my body made will still be present in my system for a very long time. No, even if I'm already okay. So, if I want to go abroad and then I want to be tested, I go to the rapid test. They will tell me I'm positive for COVID-19 even if I'm already fine because my body already contains immunoglobulin or antibodies against COVID-19. So that is correct. All right. So those are the instances no wherein rapid testing can actually be sort of more of problematic than helpful. Now there are high chances of false positives and also false negatives. But let me clarify, the process of ELISA is not prone to false positives or false negatives. No, we are only talking about false positive and false negative in terms of the urgency na kailangan nimo mahibalaan kung naa ba kay COVID-19 or wala. So that's where this comes in, diagnosis of COVID-19. But generally, ELISA, just like what you did in the SLE test, is actually very reliable. No, mahibalaan yun dayon kung naa ba or wala. Also in HIV, no, because HIV is a long-term disease mango ng HIV, dili siya diretso-diretso ba. So your body will be producing enough antibodies before you even ask to be tested for HIV. So sure gyud nga naay ka kung ma-positive siya, sure gyud nga positive ka. Or kung negative siya, sure gyud nga negative ka. But the problem with false positives and negatives is again in COVID-19 testing. Any other questions or concerns? If there are none, okay. I have my reminders here. No, the output, which is individual, is due on Saturday. That's tomorrow, no, at 12 midnight. When ang finals? The finals will be on the second week of May. So actually, if we go back to our calendar, no, okay, if we go back here, and if we finish talking about cancer next week, you have 1 week, actually around 3 weeks, kay ang last week of April, okay, wala may himuon. You have around 3 weeks to work on your final project or final output. Now, if we finish discussing about chapter 16 cancer next week. So, second week of May pa ang finals. Any other questions or concerns? None? So let me just ask a hypothetical question. If someone asks you to explain how SARS-CoV-2 infects cells, will you be able to answer them in a simple manner? Will you be confident to answer? For example, imong manghod or imong mama or papa baka nangutana or imong kung naamoy helper no si yaya nangutana sa imo. Dong, dai. Unsa day pag-affect ng COVID-19 sa ato? Ngano ato ramang baga ang maapektuhan? Nganong dili man akong mata? Nganong dili man ang akong panit makatubag mo? Will you be able to answer? The silence. I don't know if silence means yes or no. Maybe. Okay. So, um, just um, just maybe you can review this video later on if your answer is still maybe, because I think the importance of knowing these stuff is really significant with what we are experiencing right now. And of course, it's related to cell and molecular biology. So, as students of cell mol, you should be able to have the idea of how um, this works, no? Sa katong SARS-CoV-2. Okay. But anyway, um, any other questions or concerns?
Ako ano, sir, regarding sa lab report, sir ba, ang katabi
Mm.
tong worksheet sa katabi tong virtual lab, i-attach lang ba namo to sa kanang file na mo nga gibuhatan na mong lab report?
Yes, yes, just attach it to your lab report. So your lab report should still contain materials, objectives, methods in outline form, flowchart form, and then just attach the worksheet.
Okay, sir, thank you.
Okay, so wala na. 1 minute more before 3 p.m. So thank you very much, guys, now for joining in this video teleconference. Thank you for interacting, for participating. Um, if you have any more questions, no, about our topic or about other stuff in your lab report, just email me. I no, not email. Just send me a message via eLearn or through Messenger. Okay, so otherwise, if you don't have any questions, then I'm done with the discussion. Next week is our last synchronous meeting, I believe, and then finals to the sa May. So again, thank you very much. Have a productive day ahead still, and you are now all dismissed.
Thank you, sir.
Thank you, sir.
Thank you, sir.